It's my pleasure to introduce our first speaker, who is Christopher McKean. Chris, are you there? Hello.
Hello.
Great. So I'm just going to load your presentation now.
Thank you. Okay. So good afternoon, everyone. My name is Chris McKean. I'm a second-year PhD student at Memorial University of Newfoundland. And today I'll be talking to you about the taphonomic artifacts found at the Spaniard's Bay fossil assemblage in Newfoundland. Spaniard's Bay is located only an hour and a half from Memorial University and the city of St. John's, and it is unique within the Avalonian assemblage for the preservation of exquisite three-dimensional fossils, as can be seen from these images here that we took this field season. The fossils at Spaniard's Bay are typically between a couple of centimetres to 10 centimetres in length. Despite this, the level of detail they preserve is down to 0.05 millimetres, and this actually allows us to see exquisite morphological detail, such as the similar branching that is indicative of the rangeomorpha. Despite being such an amazing site, Spaniard's Bay has actually been limited in the amount of papers that have actually focused on it. Only a few papers have actually focused on Spaniard's Bay, and those that have actually tried to explain this preservation. And this has led to two taphonomic models in the past. The first of which was in Narbonne 2004, and they suggested that when these fossils were buried by a turbidity current, they were actually encased within concretions that would have protected them from both deformation and cleavage, as well as led to that three-dimensional preservation. However, both our work this field season, as well as a later study, actually showed there is no evidence for concretions at this site. And that later paper was Brazier et al. 2013. And that has the most recent and the actual currently accepted taphonomic model for this site, which can be seen actually at the bottom of this slide here. With the Brazier model, it is interpreted that the rangeomorph found at Spaniard's Bay were actually upright within the water column. And due to high currents, eddying caused obstruction scales to appear around their bases. And eventually they were actually felled into these before then being rapidly buried by a fine-grained sand brought in by a turbidity current that led to this preservation. Now, at Spaniard's Bay, there's certainly evidence for stems within these rangeomorph fronds, as can be seen from the avalofractus on the right-hand side of this slide. However, other specimens, such as the other two I've got on this slide here, don't actually have stems preserved. And what is traditionally being observed as a stem for these creatures is in fact a scour that's been generated by a holdfast that is found below the fossil. As can be seen from these images here that I have false coloured from our fieldwork this season. Here you can clearly see that you have the frond in blue, and then you have a holdfast and a scour in red. When you look at the frond, you can actually see that it is still partially buried by sediment on top of it, as is on this image on the left. And this actually suggests that what happened is the frond and the holdfast are separate organisms, and a scour occurred around the holdfast of or the stem of an organism that has the holdfast, and actually re-exposed a frond that has been buried prior to this. This can also be seen on the image on the right, where we actually have a clear dissociation between the holdfast of an organism and the frond of another organism, suggesting again that they are actually separate organisms. And finally, we actually have in the center here, some organisms that actually in the opposite direction to the paleo current. Now, this all suggests that these were actually, in fact, the fronds at least, were actually reclined organisms living on they were endobenthic reclined organisms. And as such, it's actually more parsimonious to assume that these creatures were actually growing on the surface, as opposed to this middle one here, which a previous paper actually did suggest that maybe it was felled into the current or was flipped. However, it makes more sense if these are actually creatures that were growing on the sediment surface. So this data that we've actually looked at suggests that we need a new taphonomic model. And as luck would have it, over the last few months, that's exactly what I've been working on. Now, this is still work in progress, so I'd appreciate it if we just kept this between us at the moment. But here we actually have a lot of similarities between our current model, the Brazier model, and this model. The only major difference is that the holdfast is from a stemmed organism, and the frond is from a separate endobenthic rangeomorph. This is then buried by a fine silt and begins to decay. Then, like in the Brazier model, an obstruction scour caused by an eddying current would actually re-expose this. That current would actually remove the frond or the stemmed organism, leaving the holdfast in place. And then the continued high current would actually begin to break away the slightly decayed organism, leaving the low impression, as we can see in F, where we have the remains of the organism in blue, but the impression in light brown. That is then further buried by again, like in the Brazier model, by a fine sediment, sandstone. and actually probably suggests that this is more of an amended model. Right. I only have 10 seconds left, so I'd just like to thank you guys for listening, and back to you in Oxford.
Thanks very much for that, Chris. That's really interesting. And please do keep your questions coming in the chat so we can put them to Chris at the end of the session.
Thank you.
So next we have Giovanni Pasinetti, who's here. I'm just going to start your slides now.
Hello, everybody. I am Giovanni Pasinetti, also from the Memorial University of Newfoundland. And today I'm going to talk about some exceptionally preserved rangeomorph fossils from the Bonavista Peninsula. The rangeomorphs are one of the predominant components of the Avalonian assemblage of the Ediacaran biota. They are complex sessile organisms with a seemingly fractal architecture, but despite their abundance, their phylobiology, their ecology, and their phylogeny are still widely unknown. In this work, we took in consideration the species Culmofrons plumosa, which is one of the most typical and characteristic species, and presents a frond, a stem, and a recognizable holdfast. The species was firstly described by Laflamme in 2012 from the famous Mistaken Point Ecological Reserve. But our specimen come from a different location, about 500 kilometers away from the Mistaken Point Reserve in the Bonavista Peninsula, from what's known as the moon surface, which is an exceptional surface which was discovered by Jack Matthews and Alex Liu in 2004, where the conception style preservation reveals sublime details. With this kind of preservation, a fine layer of ash would have covered the specimens on the bottom of the seafloor and preserve them as impressions on the sediment below, carrying our details of the branchings that we can see now. On the left, you can see the holotype, and on the right, you can see the specimens which I worked on from the moon surface. When we observe the frond of Culmofrons plumosa, we can see that it's composed by secondary branches organized in line, which we call primary branches, and they are further subdivided in tertiary and quaternary branches. If we take them under controlled light, we can reveal details of those branching, and we can observe that underneath certain secondary branches, there are bundled tertiary branches which appear to be furred and organized in some specific structures. The structures are located at the tip of the secondary branches, and they do not displace the neighboring branches, so we can assume that they are indeed part of the organism and not simply the organism, for example, overlying accidentally another specimen. The evidence we collected suggests that we can interpret those structures as being reproductive. In fact, the structures are consistent with the modular body plan of the organism. They are also consistent with the growth model proposed for the charnid rangeomorph, which will see them growing from the tip toward the base by addition of successive branches during the enlargement of the organism. And they do not display any evidence of damage repair, so we can assume they do not represent any kind of overcompensatory growth or damage repair structure. When we compare Culmofrons plumosa with another famous rangeomorph, Beutuchis mistakensis, we can also support a reclining lifestyle for Culmofrons. So we can assume the species was living horizontally on the seafloor, and we cannot see those potentially reproductive structures as being growing below the specimen in the sediment, and consistent with the proposed growth models. Furthermore, if we compare it with another multifoliate rangeomorph, which is Ilaluculus fordi, we can also further support the modular hypothesis of the organization of those organisms, in which the higher order branches could potentially revert to lower order branches in order to respond to potentially ecological limitation, for example, starvation strategies. In fact, when we take in consideration asexual reproductive strategies in extant animals, we can see that a lot of the times they are related to ecological limitations, and therefore they could be a response to, for example, the burial of the organism. I think these structures are really interesting because they are indeed one of the first structures that we can observe and that could give us insights on the complex and still unknown paleobiology of the group. Thank you very much.
Very much, Giovanni. That was a fantastic talk and right on time.